Hi student, I am Dr. D.S. Thaure. Now I share the screen of today's lecture in front of you. Please wait. Welcome to the next lecture after your ending of midterm exam. So course number path 354, diseases of field and horticultural crops and their management. Okay, so in today's lecture we discuss regarding the rice diseases. So this rice mainly cultivate in coastal area having high rainfall areas. So this rice crop having different diseases may be seen in including the fungal diseases, bacterial diseases and some viral diseases seen in rice crop. Okay, so in rice blast disease is the major disease seen in all the variety of the rice. So this blast occurs in all the rice growing countries in the world, causing considerable reduction in yield. Okay, so now this disease is majorly seen in all the rice growing tracts of the India, and having losses is near about 20 to 60% in grain yield. Okay, so this is the most important disease seen in all the rice growing region. Okay, so you see these photographs. So in these photographs, near about 80 to 90% rice plants having these blast symptoms are seen. Means these affected leaf and some parts of the rice plants having burning appearance are seen. Means this affected rice field showing the burning appearance symptoms. So these burning appearance symptoms generally seen due to the blast disease. Okay, so now we discuss regarding the which type of the symptoms produced on rice plant due to the blast pathogens. Okay, so this fungus attacks to the rice crop at all the stages from seedlings. Means this is majorly seen in nursery as well as to the main fields. Okay, so this pathogen develop the symptoms on leaf sheath, rashes, nodes, panicles, and also the glooms. Means all the aerial plant parts of the rice showing this type of the symptoms. Okay, so first of all we discuss regarding the leaf spot. Means these pathogens attack to rice plants and develop the different types of the symptoms. Means leaf blast symptoms, nodal blast symptoms, neck blast or panicle blast symptoms, and bloom blast symptoms. Okay, so this type of the symptoms majorly seen on the rice plants. So first of all we discuss regarding the leaf blast. Okay, means when the pathogen infect to this healthy one leaf of these rice plants, so initially small water soaked bluish green spots generally seen on the rice leaf. Okay, so these water soaked bluish green spots just enlarges and to form the spindle shaped spots. Okay, so you see these spindle shaped spots. Spindle shaped spots means just looks like as a human eyes. Okay, means the middle portion just bulbous, and end of this or both the end of these spots generally tapered. Means this spindle shaped spots generally seen on the leaf of this rice plant. Okay, so you see these individual spindle shaped spots on this leaf. Okay, so the gray center with dark brown margin spots generally seen on the leaf. Okay, means you see the individual spindle shaped spots. Okay, so these spindle shaped spots generally center points generally gray in color with surrounding the dark brown margins. Okay, so under favorable conditions or under most moist conditions, these individual spindle shaped spots coalesce to each other. So these spindle shaped spots grows on the entire leaf surface. Okay, so majorly portion of the leaf showing the dry appearance or the infected leaf takes place to the dries. Okay, means such infected leaf become dries and withers. Okay, so these pathogens mainly spread from leaf to the leaf sheath. Means same type of the symptoms also be seen on the leaf sheath portion. Okay, so when this infection seen in the nursery bed seedlings of the rice, so these affected seedlings dries up and withers. Okay, so this type of the symptoms is known as the leaf blast symptoms. Okay, then we discuss regarding the nodal blast symptoms or node blast symptoms. Okay, means these pathogens develop the symptoms firstly on the leaf surface, then leaf sheath, and finally develop the symptoms on the nodal portion of this rice plants. Okay, so initially the irregular dark brown lesions are somewhat these blackish areas seen on this infected nodal portion of these rice plants. Okay, so under favorable conditions, these blackish lesions may be encircled or may be girdle around these nodal portions. Okay, means these affected tissues become weakened. So after some days, these affected nodes may be break up and all the plant parts above the infected nodes or nodes may be dies. Means due to the heavy infection seen on this nodal portion, so this nodal portion having the tissues become weakened. So these upper parts of this nodal may be break up or broke down these upper portions. Okay, so these symptoms is known as the nodal blast. Okay, then we discuss regarding the next type of the symptoms, that is a neck blast or panicle blast. Okay, means at the flower emergence the fungus attack the pendulums or this neck portion or panicle portion of this rice. So initially dark brown lesions is seen on this neck portion or panicle portions. So these pathogens grows under humid conditions and these lesions is engirdled or maybe covered to these neck portions. Okay, means these brownish black lesions may be engirdled around this infected portion of this neck. Okay, so this neck portion or panicle portion become rotted. So this type of the symptom is known as the neck rot symptoms or neck blast symptoms or panicle blast symptoms. Suppose the pathogens attack in early neck infections, so grain filling does not occurs and panicles remain erect like as a dead heart caused by the stem borers. Means suppose these infections generally seen before the actual grain filling in particular panicles. So these panicle having no grains are formed. Only panicles are formed, but these panicles just grows in erect portions. Okay, but sometimes these pathogens attack or these pathogens develop the symptoms after the some days, means after the grain filling stage. So the infected panicles having the only few grains are infected and some grains to be healthy one grains. Okay, means these infected panicles having the glooms are somewhat infected. Okay, so these photographs, that is the neck blast or that is a panicle blast. Okay, then we discuss regarding the last blast, that is a bloom blast. Means already the pathogen infect to the panicle or neck regions. So this panicle having the grains or glooms to be infect and showing the brown to black spots. Okay, means these brownish to blackish spots generally seen on the outer surface of these glooms. So further infection goes to the up to the seeds. So these seeds become brown to black in colors. Okay, so this is the symptom generally developed on the rice plant due to the blast pathogens. Okay, understood these symptoms regarding the blast disease seen in rice crop. Okay, so you clear cut see these spindle shaped spots generally seen on the leaf of the rice crops. So several this or this type of the spindle shaped spots may be seen irregularly portion of this entire leaf blade or leaf surface of the rice plants. So finally this infected leaf generally dries from tip points to the downward directions. So in this way, number of the leaf generally dries. So burning type of the appearance or burning appearance symptom generally seen on the rice fields. Okay. So this symptoms is known as the gloom blast. Means already brownish spots seen on the outer surface of the gloom. So these glooms develop the seeds. So finally these seeds is also infect and showing the brown to black in colors. Okay. Then we discuss regarding the causal organisms. Okay. So Pyrifluria grisi, this pathogen is mainly responsible fungus to develop the blast disease in rice. Okay, so Pyrifluria grisi, this fungus develop to the asexual spores, and this Magnaporti grisi produces the sexual spores. Okay, So we discuss regarding the etiological parts of this pyrifluoria glycine and magna glycine. Okay, so this pyrifluoria glycine develop the mycelia. So this mycelia is always aligned to olivaceous and septate in manners. Okay, so this conidia generally produce in cluster on the tip of this conidiophore. Okay, so you see the individual conidia. So conidia generally pyriform to obclavate or somewhat the top shape, and this number of the pyriform conidia generally attach at the tip of this conidiophore. Okay, then you see the number of the septa, how many septa generally produced within this single conidia. Means in blast pathogens or pyrifluoria glycine pathogen develop the conidia. So inside this single conidia, generally three-celled conidia, and this conidia generally align to somewhat the pale olive green colored conidia generally seen. Okay, three septa means 1, 2 and 3. So this is the three septa conidia generally produced at the tip of this conidiophore. Okay, means conidia reproduced by asexual method by the fungus pyrifluoria glycine. Okay, then this fungus also be reproduced by sexual methods, means to develop the sexual spore fruits, that is known as the perithecia. Means perithecia, it is the flask shaped spore fruits having the asci are developed. Okay, so this asci having the 8 ascospores are developed, and these ascospores generally haline. Haline means colorless, and these ascospores generally four-celled and may be curved in natures. Okay, means this fungus reproduced by sexually as well as the asexually. Means primary infection takes place through these ascospores present in the infected plant parts or may be present inside the seeds or seed pod. Okay, and secondary infection means spread of the disease from one plant to the another plant through this conidia. Okay, so this fungus produces the few toxins, that is picolonic acid, pyrifluorin acid and pyricolol. Okay, so these three acid produced by this pyrifluoria glycine pathogen and magna glycine pathogens. Okay, so you see these photographs of the conidia of the pyrifluoria glycine. So this is the 1, 2 and 3. Means three-celled conidia is produced at the tip of the conidiophore. Okay. So these photographs, that is the conidia generally means pyriform conidia is produced at the tip of the conidiophore. Okay, so this is the disease cycle of the pyrifluoria glycine or magna glycine. Means these primary infections generally takes place from the ascospores or maybe conidia present in the infected plant part. So these spores generally landing on the healthy one host tissue. So these spores develop the germ tubes. So actually pathogens penetrate within these host cells. So these host cells having the mycelia is developed intercellularly manner. Means these spores just to produce the mycelia. So finally produces the conidiophore and conidia. So this conidia liberated from this host tissue and just deposit on the healthy one host tissues. Okay. So in this way, the primary infection and secondary infection takes place and complete the life cycle of the pyrifluoria glycine magna glycine. Okay. So this is the disease cycle. Okay. Then we discuss regarding the perpetuation and disease cycle. Already I discussed regarding the perpetuations. Means primary infection takes place, that is infected seeds is used for sowing purpose because this infection generally seen on the glume and outer surface of the seeds. So such infected seeds generally used as a sowing purpose in the next year. So 100% disease is reduced. Okay, then infected plant part debris in the soils. This is the another possibility to initiate the disease in the healthy one plants. Then in the absence of the rice crop, this fungus is also developed and overwinters in regarding the collateral host. So this is the number of the collateral host may be seen in the rice field or around the rice field. So this pathogen generally survival on this collateral host. Okay. Then secondary infection that is always takes place through conidia, spread through wind or air currents. Okay. So this is the perpetuation and disease cycle of the pyricularia vilce pathogens. Okay. Then terrible conditions. This is the excessive doses of the nitrogen fertilizers, then cloudy weathers, high relative humidity, then low night temperature, then number of rainy days, then longer duration of the dews. Means some dews generally seen or some moisture is present on the surface of the leaf, then cloudy weather. So this is the several factors to initiate the blast disease in the rice fields or rice crops. Okay, then how to control this disease seen in the rice crops? So first of all, use the disease free seeds is used for sowing purpose. Then remove and destruction of the weed host, that is a collateral host seen in the bunds or channel in the rice fields. Then collect and burn the plant debris and collateral host properly. Then treat the seeds with Captain Thyrum or carbendazim or carboxyl or tricyclazole at the rate of 2 gram per kg of seed meal to protect the seeds from the fungal infections. Then this already treated seed with fungicides. So these seeds again treated with using the biological agent that is a trichoderma viride at the rate of 4 gram or Pseudomonas fluorescens at the rate of 10 gram per kg of seeds. Okay. Then avoid close pressing of the seedlings in the main fields. Then growing resistant varieties that is Ratnagiri 68 or IR8 or Karzat 1 or IR50 or Co 39s or apply split doses of nitrogen or judicious application of nitrogen fertilizer. Suppose the farmer can apply excess doses of the nitrogen, so the blast disease initiate in the rice fields. Then spread in nursery area because this is majorly seen in the nursery seedlings of the rice. So avoid these seedlings from the blast pathogens by applying carbendazim 25 gram or idifenphos 25 ml per 10 liter of the waters. Okay. Then foliar sprays of the Bodax picture 0.5% or copper oxychloride 0.3% or idifenphos 0.1% or manib 0.25% or carbendazim 0.1% or sometimes some antibiotic substance or product may be used to recover this blast disease that is blastocidin 100 ppm or casugamycin 100 ppm at 10 days intervals for controlling the secondary spread of the disease means to eradicate the conidia. Okay, or sometimes spraying of the fluorescent pseudomonas bacterial cells at leaf and neck infections generally seen on the rice plants. Okay, so this is the several management practices adopted by the farmers. So finally, eradicate the blast disease in rice or avoid the blast symptoms in rice. Okay. Okay, now we discuss the second important disease seen in rice and produces the symptom or produces the disease, that is the brown spot of paddy. Means brown spot symptom generally seen in rice. So this disease is also known as the fungal blight or sesame leaf spot. Okay, so this disease is also occurs in all the rice countries in the world. Okay, so now this is majorly seen in coastal areas having the most commonly cultivate the rice crops. Okay, so this blast disease generally seen in seedling stage of the rice plant or in the mature plants. Okay, means these pathogens develop the symptoms on growing seedlings. So this symptom is known as the seedlings blight symptoms. Okay, means the fungus generally develop the symptoms on growing seedlings of the rice. Okay, so just we see the photographs. Okay, so this symptom generally seen in growing seedlings of the rice. So initially, just a brownish spots may be seen on the hypocotyl and epicotyl structure of just younger seedlings of the rice. So these brownish lesions may be spread in upward direction and downward directions. So these affected seedlings is totally blight in appearance. Okay, so this symptom is known as the seedlings blight symptom, and this seedlings blight symptom majorly seen in the nursery bed areas. Okay, then this fungus also be developed to the growing mature plants. Okay, so initially these pathogens attack to the leaves, leaf seed, nodal portion, panicle portion and bloom portion. Means this fungus attack to the all the growing portion of the rice plants and develop the symptoms. So initially the brown round to oval spots. Okay, so you see this brownish oval to round spots seen on the leaf of this rice plant. Means initially just isolated brownish or round to oval brown spots or brown colored spots seen on these leaf portions. Okay, so initially these spots is isolated, but under favorable conditions these isolated spots grows and enlarges to the entire surface of this leaf seed or leaf blades. So these isolated brown spots coalesce to each other and to form the large patches of the withered tissues. So you see these large patches or this blight appearance type of the symptoms seen on these leaf portions. Okay, so heavily infected leaf generally dries and defoliated from this rice plant. Okay, so this symptom generally seen on this leaf portion. Then this fungus also be developed to the nodal portion or panicle portion or bloom portions. Okay, means initially brownish spots may be seen on the stem portion or maybe nodal portion or maybe panicle portion or finally may be seen such a brownish spots on the grain portions. Okay, suppose this infection seen on the neck portion. So in this case, this infection seen as a superficial type of the infection. So these infected nodes or nodal portion or these infected panicles cannot break. Okay, but in case of the blast disease, this fungus penetrates deeply. So this infected neck or panicle so finally break. But in case of the brown spots, this infected panicle or infected neck cannot break. Okay, so this is the major difference between the blast disease and brown spot disease. Okay, so these brownish spots or somewhat blackish spots seen on this seed of the rice. Okay, so these infected seeds may be mixed with the healthy one seeds at the time of the threshing operations. Okay, so this is the symptoms develop in rice plant due to the brown spot pathogens. Okay, then we go to the which causal organism is responsible to produce the brown spot disease in rice. So this is the Helminthosporium oryzae. So this Helminthosporium oryzae fungus reproduced by asexual methods and to produce the conidia. And this Dressler oryzae, this is the perfect stage, means this fungus reproduced by sexual method and to produce the sexual spore fruits. Okay, so already we discussed the etiological or morphological characters of the Pyricularia glycine and Magna glycine in blast disease. But in regarding the Helminthosporium oryzae, this fungus is also reproduced by asexual method and to develop the conidia. So this type of the conidia, so this conidia having the several septa. So this septa may be 6 to 14 in numbers. Okay, so this 6 to 14 number of the septa is seen within the single conidia. So this conidia generally produce at the tip of this conidiophore, and this conidia generally pale olive green to golden brown in color. Okay, so this conidia reproduced by a sexual method by the fungus, that is a Helminthosporium oryzae. Then this fungus also be reproduced by sexual methods by forming the perithecium. So inside the perithecium fruiting body, asci generally produced. So this asci produces the ascospores. Okay. So this fungus produces the phytotoxins such as cochilobolin A, cochilobolin B, and opisbolin first. So these three phytotoxins produced by this Helminthosporium oryzae for easily penetrate within the healthy one host tissue. So this fungus, these pathogens, phytotoxin produced by this pathogen. Okay, so this is the morphological structure of this Helminthosporium conidia. So this is the 1, 2, 3, 4, 5, 6, 7 or 8, means 6 to 14 number septa generally produced within this single conidia. Okay. Then primary infection and secondary infections. Okay, so infected seeds, infected plant parts. So these two sources to initiate the disease in the healthy one plants. Or this fungus also be survival on collateral host, that is Digitaria or some weed species or collateral weed species seen around the rice fields. Okay, or sometimes the potassium deficiency majorly seen in a rice plant. So initiate the brown spot disease in rice plant. Okay, so this is the disease cycle of the brown spot pathogen. So this is the infected seeds sown in the fields. So 100% disease is produced and some symptoms may be seen on the younger leaf. Okay, means these growing seedlings become infected and showing the somewhat brownish spots. So these brownish spots produce the conidia. So this conidia released from this infected host tissue and just fallen on the other healthy one plants may be deposit or landing on the leaf portion or maybe bloom portion or maybe panicle portion. So this portion become infected. Okay, so these fungus produces the flask-shaped fruiting bodies having the ascospores. Okay, or somewhat this infected host tissue produces the conidia, and this conidia disseminated from this place to the this place through wind or air currents. Okay, then control measures or management practices regarding the brown spots. So use disease precedes, removal of the collateral host and infected debris in the fields, then seed treatment with the thiram at the rate of 3 to 4 gram per kg of seed, or the pre-soaking of the seeds for 24 hours in cold water, then dipping them in the hot water at 50 to 54 degrees Celsius for 5 minutes. Okay, means suppose firstly the rice seeds soak in the cold water having the temperature is near about 26 to 27 degrees Celsius. So this mycelia already survival inside the seed coat of these seeds. So this fungus become germinate and to produce the somewhat mycelia. So this mycelia just killed after such seeds deep in the hot waters. Okay, then excess or deficiency of nitrogen fertilizer increase the susceptibility of the plant. So farmers can apply the balanced fertilizer dose, that is a nitrogen, phosphorus, and somewhat increase the dose of the potassium. Okay, then spraying the crop with mancozeb at the rate of 0.3% or copper oxychloride at the rate of 0.25% or zineb at the rate of 0.1%. So applying these fungicides over these infected rice fields, so very effective control this disease by applying these fungicides. And growing resistant varieties, that is Bala, BAM10, IR20, Jaya, Ratna, then Bala Krishna, Sarbati, IR4, Patnai. So this is the some hot resistant and tolerant variety will be grown in the fields. So avoid such type of the infection in rice crop. Okay. Then after blast and brown spot, the third most important disease seen in rice, this disease is known as the sheath blight disease. So this disease majorly seen on the leaf sheath portions. Okay. So initially the greenish gray. Okay, so this is the greenish gray ovoid or somewhat ellipsoid large lesions is developed on the outer portion of the leaf sheath. Okay, so this ellipsoid or ovoid greenish gray lesions is spread along the or on the surface of the leaf sheath and maybe leaf. So these lesions may be 2 or 3 centimeter in length. Okay, so these greenish gray spots become greenish white center with dark brown margins. Okay, so this is the grayish white center spots having the brownish line. So these spots or these lesions mainly seen on the surface of the leaf and leaf sheath. Okay, but under favorable conditions or in the advanced stage of the infection, so brown sclerotia generally formed on this infected leaf sheath. Means these leaf sheath are easily pulled out because this leaf and leaf sheath is totally rotted. So these affected plants is easily pulled out. Okay, so in the advanced stage of the infection, the fungus develop the mycelia or whitish color mycelia with sclerotia body. So this sclerotia means modification of mycelium or spores. Okay, so this sclerotia generally seen inside this infected leaf sheath or underside of the leaf. Okay, so this number of the sclerotia may be formed on the infected leaf and leaf sheath. So finally this infected leaf and leaf sheath is totally rotted, and this affected leaf and leaf sheath can easily pulled out from the plants. Okay, so in severe cases, these leaf of the plants are totally blight. Means these affected plants having the several leaves become infected and showing the blight appearance. So finally these infected plants will be dies. Okay, means in leaf sheath disease, the mainly two types of the symptoms are seen. Means initially just grayish white spots with surrounding the brownish margin. So such type of the lesions are developed. But under favorable conditions, this fungus develop the sclerotia and mycelia. And this mycelia and sclerotia generally produced within this infected leaf and leaf sheath. Okay, so this is the symptoms developed on the rice plant due to the leaf sheath pathogens. Okay, okay, so this is the initial infection and this is the late stage infections. Means initially just a greenish gray lesions may be seen on the leaf and leaf sheath. So this greenish gray color spots become greenish white center with surrounding the dark brown margin. So this lesion just spread in upward direction and downward directions. Okay, so this is the sclerotia and mycelia produced within this rotted portion of the leaf and leaf sheath. Okay, so the Rhizotonia solani, this fungus produces the leaf seed disease in the rice. Okay, and perfect stage of this fungus, that is a Thanatophorus sasaki. Means suppose this infected plant parts isolate from this rice plant and just grown on the artificial media in the laboratory conditions. So after 7 or 8 days, some blackish or brownish sclerotia. Sclerotia means just mustard size fruiting bodies, or sclerotia is a hardy, compact, brown to black bodies develop after the modification of this mycelia. Okay, so this is the sclerotia. So this sclerotia initially white in color, but after few days this sclerotia may be brown or maybe purplish brown or maybe black in colors. Okay, so this is the disease cycle of the rhizotonia solani. Means this is the infected plants having the young sclerotia are seen, and this sclerotia disseminated from one place to the another place. Means this sclerotia may be float on the surface of the water. So this sclerotia may be reached to the another healthy one plants. So this infection mainly spread to the another plants. So these another healthy one plants become infected and showing the blight appearance, or somewhat the greenish gray color lesions may be seen on the leaf and leaf sheath. So this mycelia generally seen inside this infected leaf and leaf sheath, but this mycelia sometimes develop the sclerotia. Okay, so this is the disease cycle of the rhizotonia solani. Means this mycelial network or cushion shaped mycelia or mycelial network just grows inside this healthy one host tissue. So this healthy one host tissue uh, become infected. So this mycelia generally develop the symptoms on the growing rice plants, or somewhat these infected plant parts showing the somewhat these knot like structures are seen. Okay, so this young mycelium just infect to the healthy one plants. Okay, or sometime this young mycelium germinate under the favorable conditions and to produce the sexual spores, that is basidospores. Means these basidospores reproduce by sexually methods by forming the basidiocarp. So basidiospore generally produced by the Thanatiphorus sasaki fungus, and this type of the mycelia generally developed by the Rhizotonia solani fungus. Okay, then perpetuation means the sclerotia, generally fallen or infected plant parts having the sclerotia, so debris in the soil, so 100% disease is produced from this sclerotia is survival for several months in the soil or in the infected plant parts, and secondary infection takes place through sclerotia. So this sclerotia may be float on the surface of the water, so finally reach to the another healthy one plants. So this healthy one plant become infected. Okay, then control measures: avoid flow of irrigation water from infected field to the healthy fields, because the secondary infection or secondary spread of the disease through sclerotia. So this sclerotia mainly float from on the surface of the waters. Then deep flowing in summer and burning of the stubbles, because this sclerotia may be survivable in soil for a longer durations, means up to 7 to 8 years. So deep flowing practice is generally done. Then seed treatment with Pseudomonas fluorescens at the rate of 10 gram per kg of the seeds, followed by seedlings deep at the rate of 2.5 kg Pseudomonas fluorescens per hectare dissolved in 100 liters, and finally dipping for 30 minutes. Means to avoid the pathogens or avoid the attack of the pathogens to the younger seedlings. So these protect the seedlings uproot from the nursery bed areas. So these uprooted seedlings from the nursery bed area, so immediately dipped in the prepared solution by using these Pseudomonas fluorescens. So you can control the disease. Then soil application of Pseudomonas fluorescens at the rate of 2.5 kg per hectare after 30 days of the transplanting. Okay, so this Pseudomonas fluorescens powder that is a 2.5 kg powder mixed in the 50 kg FOIM. So this FOIM is broadcast on the rice field. Then soil drenching with the kitazine or edifene phos or benomil at appropriate concentration, that is 0.1 to 0.2% during the tillering stage. So you can avoid the infection to the leaf and leaf seed. Or spray the propiconazole at the rate of 0.1% or hexaconazole 0.2% or validamycin. So this validamycin is a antibiotic product generally applied at the rate of 0.2%. Foliar sprays at 0.2% concentration from 45 days after the transplanting at 10 days interval for 3 times. Okay, so in this way farmer can avoid the infection of the Rhizoctonia solani to the rice field and produce the Rhizoctonia blight or seed blight disease. Okay, then last disease, this is a not disease. This is the symptom produced in rice field due to the deficiency of the zinc. Means zinc deficiency generally seen in the rice field. So 100% this type of the symptoms are seen. Okay. So this is known as the Khaira disease. Khaira disease of rice. Okay. So this is the abiotic disease, not a biotic disease. This type of the symptom is not caused due to the any pathogens. Okay, so this is the abiotic disease. So this abiotic disease generally seen due to the several factors. Suppose the farmer can growing the rice in alkaline and sodic soil. So 100% disease is produced. Then sometime the farmer can done the intercultural operations, that is soil leveling. So these expose these subsoils at the time of the leveling. So 100% this type of the symptoms are seen. Then if well water has calcium carbonate, means suppose this well water or bore water having the high percentage of the sodium carbonates, sodium bicarbonates, so 100% this is or this type of the symptoms are seen. Okay, then the farmer irrigate with the using sewage water. So this sewage water having the higher percentage of the phosphate. So these sewage water generally used for irrigation purpose. So 100% this is produced. Then this Khaira disease mainly seen in the calcareous soils with high organic matter content or soil with higher available phosphorus. Okay, so this is the several factors or several abiotic factor to be considered for showing the zinc deficiency or showing the this type of the symptoms in rice crop. Okay, so now we discuss regarding the symptom. So initially these symptoms usually appears or seen from 2 to 4 weeks after the transplanting or sowing. Means majorly these symptom generally seen in the younger seedling. So initially these affected younger seedlings having the leaf, so this leaf generally yellow in color. Means these chlorasis symptoms or chlorotic lesions generally seen at the basal side of the leaf of this growing plants. Okay, so this chlorotic leaf generally seen from the basal side of the leaf. So later stage, this chlorotic leaf is also seen on the top portion of this plant. Means this chlorotic leaf exceed from basal side to the lip tip. Okay, so initially reddish brown to brownish irregular lesions or blotches appears on the leaf blade. Okay, so you see the infected leaf and healthy one leaf. So the affected leaf blade is always smaller as compared to the size with the normal leaf seed. Okay, so this affected leaf blade having somewhat the reddish brown spots are seen. Means bronzing type of the symptoms generally seen. Okay, means in case of the severe deficiency, entire leaf may use the rusty brown appearance. So this symptom is also known as the thyroidosis. Okay, means somewhat the reddish rusty brown lesions seen on the leaf. So this symptom is known as the thyroidosis. Okay, so in case of the severe deficiency, maybe zinc or other abiotic factor, so plants will be dies. Okay, so these symptoms may be more pronounced during the early growth stage of the rice stage because zinc immobilizations. Okay, means zinc immobilization means due to the increased bicarbonate concentration in the soil under strongly reducing the condition following the flooding. Means during the flooding or some flooded water seen in the rice field. So bicarbonate concentration is increased in flooded waters. Okay, so these plants is a totally dies or this plant having the leaf having the rusty bronzy type symptoms. Okay, so this deficiency may be cured or may not be cured. Okay, suppose the heavily plants infect by the some abiotic factors, so this plant cannot be recovered. But the deficiency is not severe, so these affected plants can recover after the 4 to 6 week. Okay, but these plants delay the maturity, that is a grain maturity, and also yield is reduced as compared to the healthy one plants. Okay, so this is the symptom generally seen due to the thyroid disease. Okay, so this thyroid disease generally seen in the rice crops due to the some abiotic factor and deficiency of the gene. Okay, so how can manage or how can reduce or recover this deficiency in rice fields? Okay, so first management point, that is a grow zinc efficient varieties. Means apply the zinc sulfate to this rice field. Then use fertilizer that generate the acidity. Means replace some urea with ammonium sulfate. Then apply organic manure before seedlings or transplantings, or apply to the nursery bed areas few days before the transplanting. Then allow permanent flooded areas to drain out and dry out periodically. Then dip the seedlings or pre-soaked seeds in a 2 to 4% zinc sulfate solution or suspension, or apply 25 kg zinc sulfate per hectare before transplanting or sowing. So repeatedly you can apply this zinc sulfate after the 3 seasons in the fields of the known deficiency, zinc deficiency plot. Okay, then this deficiency can be corrected by the dipping seedlings roots in the 2% zinc sulfate suspension for 10 to 15 minutes. Means to recover the zinc deficiency in seedlings by applying the zinc sulfate solution, or foliar sprays with 5 kg zinc sulfate plus 2.5 kg slacked lime in 1000 liter of the water per hectare. So first spray should be applied 10 days after the sowing in the nursery bed area. Second spray should be applied 20 days after the sowing, and third within the 15 to 30 days after the transplant. So in this way, you can manage the Thera disease in the rice crop. Okay, so you can understood the blast disease, brown spot disease, seed blight disease, and Thera disease in the rice. Okay, so this is the disease majorly seen in the rice crop. Okay, so same type of the symptoms or same type of the disease also be seen in the finger millet or ragi, that is a blast disease. So this is the symptom generally seen on the ragi plant caused by the pyricular ragis and produces the blast disease. And second most important disease, that is a leaf spot disease, that is caused by the helminthosporium citarium. So this information is similar to the blast and brown spot disease in the rice. So you can study these diseases of the ragi. Okay. So any doubt in today's lecture, please ask me. Okay.